everyone, in this video we're going to be dealing with protein synthesis. We're going to talk about gene expression and RNA, we'll look at the different stages of transcription and translation, and we'll talk about the genetic code as well. So what do we mean by gene expression? Well, gene expression refers to the display of genetic information in the phenotype. So seeing that information that is locked away in the DNA outside as a trait. And gene expression can be summarized into 3 steps: transcription, translation, and then protein activity. So during transcription, what happens is we take the information in DNA and it's converted into RNA, specifically messenger RNA. Then during translation, what happens is that messenger RNA is read and that information is converted into a strand of amino acids to form a protein. And then in the last stage, what happens is we take that protein and that protein is functioning and producing the trait that we're seeing. So let's talk about the following analogy. Let's pretend that a gene is like a recipe in a cookbook and the protein is like the baked product that you're going to be making. So let's say that your gran handed down to you this amazing recipe book with tons and tons of recipes, but the recipe book's very bulky, it's very big, and it's very fragile. So you don't want to constantly be taking it into the kitchen and using it. You don't want to mess it up. So you leave the recipe book in your library. And when you want to make a specific recipe, you go to that library and you open up your recipe book and you find the recipe that you're interested in. And what you do is you just copy down that recipe onto a piece of paper. Now that piece of paper is easy to move around with. So you take that piece of paper from your library, you move into your kitchen, and you use that recipe to make your baked goods. That recipe is going to tell you exactly what ingredients you need and in what order to add them to one another. And finally, you'll produce your baked good. So this is like transcription and translation. Your recipe book is like your DNA. You don't want to be carrying it around the cell. It's big, it's bulky, and you need to protect it. So you leave that DNA in the nucleus. But when you want to use a specific gene within that DNA, you make a copy of that gene in the form of messenger RNA. Now that messenger RNA is able to freely leave the nucleus, it'll go into the cytoplasm, and that's where translation is going to occur. You're going to take that information in the mRNA and convert it into a protein. The mRNA is going to tell the cell exactly what amino acids to add and in what order to add them. So let's talk about the similarities and differences between RNA and DNA. Firstly, looking at their similarities. They're both nucleic acids, and the monomers of nucleic acids are nucleotides. If we look at the differences, however, we notice that the sugar in DNA and RNA is different. Even though it is a pentose sugar in both, in DNA we have deoxyribose, whilst in RNA we have ribose. If we look at the nitrogenous bases, DNA is going to have guanine, cytosine, adenine, and thymine. RNA, on the other hand, does not have thymine. Instead of thymine, it has uracil. So the 4 nitrogenous bases we find in RNA are adenine, uracil, guanine, and cytosine. Our DNA is going to be a double-stranded molecule, whilst an RNA is going to be a single-stranded molecule. DNA is also far more stable than RNA. DNA is designed to last the entire lifetime of the cell. It has to last. It's got all the information in order for that cell to function. RNA, on the other hand, is made and used to make protein and then is degraded down back into its original building blocks so that those building blocks can be used to make a new strand of RNA so that a new gene can be transcribed. When we're talking about the production of protein, we have 3 main types of RNA involved. We have messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA is used to make a copy of the DNA. So the DNA is going to be read and it's going to be transcribed into messenger RNA. That messenger RNA will then leave the nucleus and move to the cytoplasm. The messenger RNA instructs the cell what amino acids to add and in what sequence to add them. This is done by reading that messenger RNA in sets of 3. Three sets of nitrogenous bases on a messenger RNA are known as a codon, and each codon will code for a specific amino acid. The ribosomal RNA is a major component of our ribosomes, and this ribosomal RNA is going to be involved in joining our amino acids together, forming those covalent bonds. The transfer RNA is going to be able to read that codon that we find on the messenger RNA, and by reading that codon, it's going to bring in the correct amino acid to add to the sequence of amino acids. Alright, so let's first have a look at transcription. So transcription is where we're taking the information in DNA and converting it into messenger RNA. Think of it as if you're transcribing something, you're remaining in the same language. So we're remaining in the language of nucleotides. However, when we get to translation, if you translate something, you change from one language to the other. So in translation, we change from the language of nucleotides to the language of amino acids. Alright, so during transcription, we're going to take that information in DNA and we're going to make a copy of it in the form of messenger RNA. This is going to take place inside the nucleus, and then that messenger RNA can exit the nucleus and move to the cytoplasm for translation. Our transcription is going to take place in three stages: initiation, elongation, and termination. So let's have a look over here. We have the sequence of bases in our DNA, and those sequence of bases are going to be coding for a specific protein. We call those sequence of bases the gene. In front of the gene, we'll have a gene promoter. This gene promoter is indicating to the cell where the gene starts. On the end of the gene, we have what we call the terminator, and this sequence of bases is indicating to the cell where that gene ends. So during transcription, we have a special enzyme involved, and this enzyme is known as RNA polymerase. And during initiation, the first thing that happens is that RNA polymerase finds the gene promoter and it binds to it. Then we move into elongation. During elongation, the RNA polymerase is going to move along that template strand of DNA. And as it does that, it's going to be producing our messenger RNA, adding one nucleotide at a time. It's going to read the sequence of bases in that DNA strand, and it's going to add a complementary nucleotide in the mRNA strand. It's going to use those same base pairing rules that we find in DNA. So where we have a T in the DNA, we will now have an A in our mRNA. Another T means we have another A. Where we have an A on the DNA, remember that we don't have thymine in RNA. Instead of thymine, we have uracil. So whenever there is an A in the DNA, it means that in the RNA, we will have a U for uracil. Once the RNA polymerase reaches the terminator, transcription is going to stop. The piece of DNA will be released, the RNA polymerase will be released and can be reused, and the messenger RNA will be released and then moves into the cytoplasm for translation. Alright, let's have a look at this question. What is the complementary RNA sequence to ATGC? Well, if we have a look at our answers, the answer is B. Where we have an A, we would have a U because we don't have thymine inside messenger RNA. Where we have T, we would have A, G, we would have C, and C, we would have G. Alright, so as I pointed out earlier, three sets of bases on the messenger RNA will be a codon, and each codon will code for a specific amino acid. So when we talk about the genetic code is describing all the possible different codons we have and what amino acids they are coding for. The genetic code also includes the start codon, which tells the cell where to start translation, and then stop codons, which tells the cell where to stop translation. The genetic code is unambiguous, which means that each codon codes for one specific amino acid. The genetic code is also redundant, and this means that some amino acids are coded for by more than one codon. So we have 64 possible codons, but there are only 20 amino acids. The genetic code is virtually universal, which means that nearly all organisms are using the same genetic code. So they're taking their DNA and they're reading it the same way that our body cells would read that information. This is why we can take human genes and we can place them, for example, inside bacterial cells, and those cells can produce specific human proteins for us. So if we just have a look at this diagram over here, we have our template strand of DNA, and the first thing that's going to happen is that DNA is going to be transcribed into messenger RNA inside the nucleus. This messenger RNA will then move to the cytoplasm where translation will occur. And that messenger RNA is going to be read in sets of 3 bases. Each 3 bases represents a codon, and a codon is going to tell that cell what amino acid to add. So for example, here we have AAG, and that codon symbolizes lysine. So this table highlights all 64 codons with their amino acids that they code for. You can see once again that we have some amino acids that are coded for by more than one codon. But if you have a look at each codon, they're only coding one amino acid. We also have our start codon, which is AUG. And AUG codes for the amino acid methionine. So often at the starting point of most proteins, we will find this amino acid methionine. We have 3 possible stop codons, and those tell the cells where to stop translation. Okay, so let's talk about the next stage, translation. Translation is going to occur in our cytoplasm. It's going to require messenger RNA. It's going to require our ribosomal RNA, which is inside the ribosomes. And it's going to also make use of transfer RNAs. So during translation, the information that we find in messenger RNAs is going to be converted into a polypeptide. So into a chain of amino acids linked together. And once again, this is occurring inside our cytoplasm. In order for translation to occur, we need the help of transfer RNA. Now, transfer RNA has what we call an anticodon, and that anticodon is going to recognize the codon that we find on the messenger RNA. So let's pretend that this is our transfer RNA, and the anticodon on this specific transfer RNA is UAC. Once again, our same base pairing rules are going to apply here. So U and A will recognize each other, and G and C will recognize each other. So this UAC would recognize AUG on our mRNA. Each tRNA has a unique anticodon, and each tRNA carries a different amino acid. This tRNA, which has the anticodon UAC, is carrying the amino acid methionine. When translation occurs, the ribosome is going to scan the mRNA until it finds that start codon AUG. When that happens, our very first transfer RNA is going to come into play. The next thing that will happen is a transfer RNA that can recognize the codon ACC will bring its amino acid. So if this transfer RNA is recognizing the codon ACC, that means it must have the anticodon UGG. It'll be carrying a specific amino acid. These 2 amino acids will then be linked to one another using a covalent bond. The ribosome will then move one codon to the right. When that occurs, this transfer RNA will be released, and a transfer RNA that recognizes GTT will be brought in, once again carrying an amino acid. This amino acid then gets linked to the growing amino acid chain. This continues until a stop codon is reached. A stop codon does not code for an amino acid, and when a stop codon is reached, the ribosome releases the mRNA as well as the growing polypeptide chain. The polypeptide chain will then fold up into its tertiary structure, forming the protein. Okay, so let's answer these questions. What is the start codon? Well, the start codon is AUG, and remember that start codon codes for the amino acid methionine. What is the anticodon that corresponds to the mRNA codon GUA? Remember, the anticodon is going to be found on our tRNA, and we use our same base pairing rules. So where we have a G, we would have a C, U, we would have A, and A, we would have U. So that anticodon would be CAU. Alright, so if you can go ahead and just pause your video, answer these questions, and then come back when you're ready and I'll go through the answers with you. So firstly, what is gene expression? Well, gene expression is where we take that information that we find in DNA and we convert it into a phenotypic trait. Secondly, what are our 3 steps in gene expression? Well, firstly, we had transcription, then translation, and then we had our protein performing its function, which is bringing about that physical trait that we're observing. Explain the first 2, so transcription and translation, with regards to their location, their templates used, and then their products. So firstly, talking about transcription. This is going to take place in the nucleus, DNA is going to be the template, and messenger RNA is going to be the product. Then for translation, this is going to occur in our cytoplasm, mRNA is now the template, and the product is going to be our protein, or our polypeptide chain. Thanks for watching. I hope you enjoyed this video and that you found it helpful and interesting.